Thank you, Dr. Bell, for your kind introduction. And I also would like to thank organizers for this great opportunity. It's an honor for me to be here. Today, I'd like to talk about a Physiome study on the role of calcium interaction between organelles. First of all, what is Physiome? The word Physiome consists of physio, life, plus ome as a whole. The Physiome is a study to provide quantitative description of physiological dynamics. To achieve this, we repeat cycles of experimental analysis and mathematical model analysis. For example, we perform physiological experiments such as characterization of ion channels and transporters and measurement of ion dynamics. Then we input these experimental data into comprehensive cell model to analyze theoretically the mechanisms and to make working hypotheses. Next, we go back to experimental analysis again to validate the hypothesis. By repeating these cycles, we can gain insight into mechanisms underlying complicated phenomena. Our major interest is the role of mitochondrial calcium dynamics. Mitochondrial calcium is not a static factor. It's a dynamically changing factor. It's involved in energy metabolism, cytosolic calcium signaling, and cell death pathway. And our final goal is to integrate and reconstruct these complicated phenomena onto a computer to understand the logic. In the present study, I'll show you our recent findings of the role of mitochondrial calcium dynamics in the heart, as revealed by a Physiome study. The heart is continuously working to pump blood around the body, and this activity is driven by ATP hydrolysis. However, a human heart contains only about 0.7 grams of ATP, which may sustain pumping for about 10 beats. In addition, the cardiac workload changes dynamically from resting to heart exercising. Here, myocardial oxygen consumption rate, MVO2, is an indicator of mitochondrial ATP production. This increases in proportion to rate pressure product, RPP, which is an index of workload. At the same time, ATP concentration in the heart is kept constant. This means that mitochondrial ATP production is strictly regulated to meet the demand. In cardiomyocyte, ATP is consumed mainly by myosin ATPase, sarcoplasmic reticulum SR calcium pump SACA, plasma membrane sodium potassium ATPase, and plasma membrane calcium pump PMCA, whereas ATP is produced mainly by oxidative phosphorylation. One important metabolic intermediate is NADH, which connects metabolism and oxidative phosphorylation. And it's reported that NADH level is kept relatively constant with various heart rate. In mitochondria, NADH is produced from NAD+ by mitochondrial dehydrogenases, some of which PDHC, OGDH, and ICDH are activated by calcium. Therefore, calcium has been proposed as a possible regulator for cardiac energetics. In fact, during workload increase, such as by pacing, mitochondrial calcium and NADH dynamically change. This is from Brandes and Barr's 2002. When stimulus frequency is increased from 0.25 Hz to 2 Hz, mitochondrial calcium gradually increased. Accordingly, NADH level initially decreased, but later increased. The determinants of mitochondrial calcium dynamics have been identified in the last 15 years. For the influx, mitochondrial calcium uniporter, MCU, and its accessory proteins. For the efflux, mitochondrial sodium calcium exchanger, NCLX, and very recently reported TMM65, and proton calcium exchanger, reticulum or TMBIM5. I'll show you an interesting report by Long et al. 2015, which is on the cardiac specific knockout mouse of MCU, which is a calcium influx uniporter. The isoproteinol-induced tachycardia increased NAD plus NADH ratio in the knockout heart. Please note that the increase of this ratio means NADH decrease. This suggests that the mitochondrial calcium dynamics does contribute to cardiac energetics. Then a question arises: how do mitochondrial calcium dynamics impact on cardiac energetics during workload transition? We've got some answers and show you as today's two topics. The first topic is on the spatial distributions of mitochondrial and SR calcium handling proteins in ventricular myocytes. In 2016, De La Fuente et al. Reported that MCU distribution is biased towards the mitochondria SR interface. Here, MCU signal is closely localized with SR calcium channel lyosin receptor. The 50% of MCU is localized near lyosin receptor. They subsequently found that the calcium uptake hotspot, which is composed of MCU and lyosin receptor, lacks calcium extrusion via mitochondrial sodium calcium exchanger, NCLX, to optimize signaling efficiency. In order to test whether there is a spatial interaction between NCLX and SR calcium pump, SACA, we performed immunofluorescence analysis using mouse ventricular myocyte. First, we found that NCLX is localized to the edges of mitochondria. The green signals derived from NCLX showed striped pattern in between red mitochondrial signals. Please see that peaks appears alternately, and also see these 3D reconstructed images using Imaris software. Then we performed super resolution imaging of NCLX using NStorm. In this case, NCLX signals are red, and SACA, lyosin receptor, or sodium potassium ATPase signals are in green. These yellow rectangular regions are merged and shown here. The co-localization analysis showed that the Pearson's coefficient and Manders coefficient are the highest for NCLX and SACA combination. And 70% of NCLX is localized near SACA, and 30% of SACA is localized near NCLX. Then, what are the roles of these spatial distributions in cardiomyocytes? We have some data on SR calcium dynamics in HL1 cardiomyocyte, which is a cell line derived from mouse atrial myocytes. When we block mitochondrial sodium calcium exchange activity, NCX-MYT, by CGP37157, the SR calcium reuptake rate after the caffeine application was decelerated, shown as blue symbols. This means there is also a functional interaction between NCLX and SACA. Then we modeled these interactions. And we modeled this uneven distribution of the proteins. That is, 70% of NCLX is placed near SACA, and 30% of SACA is placed near NCLX. In addition, we placed 50% of MCU near lysine receptor. Then we incorporated this uneven model into our HL1 cardiomyocyte model and applied caffeine protocol. This uneven distribution model well reproduced the experimental data. That is, NCX MIT reduction decelerated the SR calcium reuptake rate after the caffeine application. Next, we assumed even distribution, then applied caffeine protocol. Interestingly, NCX MIT reduction hardly affected the SR calcium reuptake rate. In other words, even distribution model fails to reproduce the experimental result. This is a summary of the first topic. In ventricular myocytes, distribution of mitochondrial and SR calcium handling proteins is uneven, which may create calcium crosstalk between these organelles. Then we analyzed the role of these spatial distributions in cardiac energetics during workload transition. We constructed an integrated model of human ventricular cell with excitation, contraction, mitochondrial energetics coupling. As a base model, we chose a human ventricular cell model by Grandi et al. 2010. And into this model, we incorporated our mitochondria SR calcium interaction model, contraction model by Sim et al., and our detailed mitochondrial energetics model, in which each component are formulated according to the experimental data on substrate dependencies. These are the basic characteristics of the model. This integrated model well reproduces the experimental data on frequency dependencies of forces and ion concentrations using human cardiac muscle. Then we analyzed the NADH change during workload increase by exercise. During exercise, oxygen demand is increased by increasing heart rate, afterload, preload, and contractility. In our model, we set up the exercise protocol by increasing heart rate from 60 to 180 beats per minute, by increasing preload, stretching half sarcomere length from 0.95 to 1.05 micrometer, and by applying beta adrenergic stimulation, simply changing these factors shown here. The exercise application gradually increased mitochondrial calcium in our model. Accordingly, NADH level initially decreased, but later increased, comparable to the experimental data. Please note that the contractile force increased in our model, but not in rat ventricular trabecular, reflecting the species differences of the staircase in addition to the effect of preload increase and beta adrenergic stimulation. Then we focused on this biphasic change of NADH. The NADH level is balanced by the consumption via complex 1 of electron transport chain and by production via these 4 dehydrogenases, some of which are activated by calcium. When calcium regulations are silenced at this point, NADH change shifted downward, suggesting that the calcium regulation contribute to the later NADH increase by increasing production. Then we analyzed which intervention has the greatest effect on NADH and mitochondrial calcium. We applied each intervention one by one to the model and applied exercise protocol. And the heart rate increase and preload increase and beta adrenergic stimulation is an intervention. And it's clear that the heart rate increase is a primary intervention that perturbs NADH and mitochondrial calcium, which is comparable to the well-accepted concept that heart rate is a major determinant of oxygen demand during exercise. In addition, please see the difference between continuous and dashed lines, reflecting calcium regulation on and off simulations, respectively. The calcium regulation is apparent when heart rate is increased, but not when preload increased or beta adrenergic stimulation is applied. Finally, we analyzed the role of NCLX and SACA and MCU lysine receptor interactions in calcium regulation during exercise. We systematically varied the fraction of NCLX co-localized with SACA and fraction of MCU co-localized with lysine receptor, then applied exercise protocol to the model. For calcium regulation on and off simulations, NADH recovery from the initial drop, A and A dash, to the end of the exercise, B and B dash, are calculated, and contribution of calcium regulation is conducted according to this formula. This is a heat map of the contribution of calcium regulation. It suggested that the MCU and lysine receptor interaction determines the contribution of calcium regulation, and NCLX and SACA interaction plays a minor role. To our surprise, the contribution becomes nearly maximum with experimentally observed distributions. That is, 70% of NCLX is near SACA, and 50% of MCU is near lysine receptor. This is a summary of the second topic. Uneven distribution of mitochondrial and SR calcium handling proteins maximizes the contribution of calcium-dependent activation mechanism. The conclusion is the orchestration of calcium handling proteins in mitochondria and SR contribute to the efficient NADH supply to keep up with the increased energy demand during workload transition. I thank these past and current colleagues and collaborators for this work, and this work was supported by these grants. This April, I moved to Okayama University. I welcome young scientists. If you are interested in our work, please contact me. Thank you for your attention.
Thank you, Dr. Takeuchi. Are there any questions from the audience?
Thank you very much for a wonderful talk. In the first part, I'm really interested in the uneven distribution of the calcium-regulating molecules such as MCU and NCLX. Can you tell us the molecular mechanism for the uneven distribution?
I don't have any data, or I don't know any.
Or any approach are you planning to do?
Well, I'm interested in, but currently I don't have any. I don't have any plan to do.
Thank you.
Jamie? Hold on, Jamie. I think it needs to be switched on.
Thanks for a very interesting talk. You've shown there's clearly this sort of relationship between having co-localized mitochondrial SR calcium regulation. In your model, can you include the distance of separation? And the reason I ask is that in heart failure, you start to get sort of, changes in the shapes of the SR and T tubules, and they sort of separate. And so is that going to decrease this coupling and decrease the efficiency?
Do you mean in our model? The distance between the
Can you include distance separation in your model?
Right now, we don't have any distance data. We assume some subcompartment in the model. But right now, for NCLX and SACA part, we don't assume any subspace. We just set the calcium extrusion from NCLX directly goes into SR. It's too extreme assumption, but where in the future, I know that we have to put some distance information into our model.
Thank you.
Any more questions from the audience? Come on, you don't want to rely on my questions again. Someone. Okay, down to me. Obviously, MCU and the ryanodine receptor are closely working in concert. Are there, I'm sure there are, but of the mutations within ryanodine receptor and MCU, would they be, giving you more information on some of these pathways?
Yeah, we don't put any information on the mutation in our model, but yeah, of course I want to do, and I want to simulate the pathophysiological conditions, not only the normal conditions, physiological conditions. So that's my future plan.
Because presumably, almost as you're suggesting, those mutations may be leading to pathophysiological conditions, essentially disrupting this energy pathway that you've described so nicely here. Thank you.
Thank you.
More questions? Okay, let's move on. Thank you, Dr. Takeuchi.